Hey guys, it's Mrs. Olenichek, and today we're going to talk about life and where it came from. Maybe. We're going to start with the cell theory. So we all remember this, right? All living things are made of cells. The cell is the basic unit of structure and function in those living things. And all cells come from pre-existing cells. Now, we do have to ask, how do we know that all cells come from pre-existing cells? For a long time, people believed that cells came from nowhere. They spontaneously generated. And they had observations that supported those ideas. Along comes Pasteur, Louis Pasteur, and he decides to do an experiment to test this idea of spontaneous generation, that cells spontaneously arise. He set up two experimental groups. He had identical flasks that he filled with nutrient broth, kind of like chicken soup. He then stretched the necks of these glass flasks and bent them into S shapes, which would prevent contamination from entering. He then heated each flask to kill all the existing microbes. For one group, he broke the neck off, exposing it to the air, and the other he left intact. Now you can imagine, only the flask with the neck broken showed growth. Now this makes sense to us, but this was a big deal. By doing this, he rejected the theory of spontaneous generation and supported the idea that contamination, something in the air, was needed for that growth. The organisms didn't spontaneously generate just because the broth was there. Okay, so now we know there's evidence to support this idea that all cells come from pre-existing cells. But where did the first cell come from? That whole chicken egg idea. There's lots of ideas about where that first cell could come from. Some of them, one of them is extraterrestrial origin. The idea that the original source of some of the organic materials needed for life actually came from meteorites striking early Earth. This is testable to some extent. There is some evidence to support the idea that some carbon or organic materials on the planet did come from meteorites. And then the idea that life spontaneously arose from inorganic molecules. Again, you can test whether this is a probable explanation. The first step is to get basic organic molecules. So early Earth didn't have organic molecules. It didn't have molecules that combined carbon, hydrogen, and oxygen. There were no amino acids. There were no sugars. It was a very low oxygen environment. And we know this by looking at rock formations and the oxidation in the rocks. Now, in 1920, there was this reducing atmosphere hypothesis, the idea of the atmospheric conditions and whether or not life, or at least organic molecules, could arise under these conditions. In 1953, Miller and Urey decided to test this hypothesis. So they set up this closed system and tried to reproduce conditions of early Earth. And when they tested the water in their system, they were actually able to form organic compounds. This was a big deal. So they produced things like amino acids, hydrocarbons, nitrogen bases, and other organic molecules. This didn't necessarily prove that that's how life started, but it did support the idea that it was possible to create organic compounds in an inorganic environment without other living things. And that's one of the first steps. Before you can make cells, which are relatively complex, you have to have organic molecules or the building blocks for those cells. And so it would make sense that you'd have to start by building monomers, things like amino acids, sugars, lipids, and nucleic acids. Those monomers could then form polymers, So they would bind with each other, forming proteins, complex carbohydrates, lipids, and nucleic acids. The next step would be some mechanism for keeping your inside in and your outside out. So if you're building all these polymers, that's a form of metabolism, breaking them down, building them up. You'd want to keep those to yourself. Now, if some of these polymers had hydrophilic and hydrophobic ends, kind of like phospholipids, if they were in a watery environment, which they probably were, they would naturally form these lipid bubbles. This is important because it does separate your inside from your outside. This allows for metabolism and gives the option for reproduction when those cells divide, or those protocells divide. At this point, it's not a living thing, it's just a bubble with some stuff in it. But we're on the right track, and you can see how these would be necessary before you could get more complex organisms that showed properties that we associate with life. Things like accurate replication and metabolism. Neither can exist without the other, and both are required for at least what we define life as now. The last step would be genetics. You'd need some sort of genetic material so that you could pass on these ideas from one generation to the next. This makes natural selection and evolution possible, which leads to the diversity we see today. And so once all three of these things happened, we can see the possibility of life evolving. Now let's look at that origin of genetics. If you remember protein synthesis, DNA is transcribed into RNA, and RNA is translated into a protein. Now each step requires enzymes, which are proteins. So you need DNA to make proteins, but you need proteins to make DNA. And so we run into an issue when we start thinking about genetic material. If you didn't have DNA, you couldn't make the proteins, and if you didn't have proteins, you couldn't make the DNA. But RNA actually can perform a lot of the same tasks that DNA and proteins perform. It can serve as information storage, it's self-replicating, and it can act like an enzyme. It acts like a catalyst. It forms those three-dimensional structures just like a protein. And so currently the belief is that RNA was the first genetic material. It was later replaced with DNA because DNA is more stable. So now we come to LUCA, the last universal common ancestor. This is probably what the first living thing looked like. It was simple, prokaryotic, no membrane-bound organelles. It probably looked a lot like modern-day bacteria. So if life originated 3.5 to 4 billion years ago, what happened from there? About 2.7 billion years ago, we find evidence of free oxygen. Now this wasn't existing before. So LUCA, our ancestor, was most likely anaerobic, single-celled. Once photosynthesis evolved, oxygen in the atmosphere increased. This would have killed off most of the organisms alive at the time, but it opens the door for aerobic cellular respiration, which is actually more efficient. You get more energy from your food, and that allows for more evolution. About 2 billion years ago, we see evidence of the first eukaryotes. And so now we're going to look at how those eukaryotes could have evolved. So we know, or we assume, that our first ancestors looked a lot like bacteria today with no internal membranes. If you have these internal membranes, these folding in of membranes, you can start to form structures like the endoplasmic reticulum and the nucleus. And this creates microenvironments internally. The advantage is you can increase efficiency by specializing, just like our organelles do now. This allows for natural selection. If you're more efficient, you survive better, you leave behind more offspring, you increase in the population. Now we get to things like mitochondria. So you have this cell that has some internal membranes, this ancestral eukaryotic cell. Let's say it engulfs an aerobic bacteria, but instead of digesting it, it lets it just hang out. It's a mutually beneficial relationship. This bacteria is able to utilize nutrients more efficiently than that original cell. And it also gets a place to live. Natural selection again. These organisms were better able to survive in the environment and reproduce, and so we see many more eukaryotes now. Now, what if that happened twice? What if that original cell that engulfed a mitochondria, or what is now a mitochondria, also engulfed a photosynthetic bacteria? Again, not digesting it. Mutually beneficial relationship, natural selection occurs, and we have our plant cells, cells with both mitochondria and chloroplasts. This idea is the theory of endosymbiosis. Some of the evidence that supports the theory are that both mitochondria and chloroplasts have their own circular chromosomes, or DNA. They can actually use that DNA to make proteins. They have 70 S ribosomes, which are the same size and shape as those ribosomes seen in modern-day bacteria. They can self-replicate, and they're similar in size and shape to modern-day prokaryotes. This theory was proposed by Lynn Margulis. At the time, she got a lot of pushback, but there's been more and more evidence to support this idea that modern-day eukaryotic cells evolved from prokaryotes that engulfed other prokaryotes, and they decided to live together in a symbiotic relationship. So here is a list of the IB understandings and applications that we've discussed. You should make sure that you can complete your BILS student objectives. If you have any questions, let me know. I'd be happy to go over them in class with you. Have a good day.